It's my pleasure to introduce Francisca Michor, my co-PI on our U54 Center grant. Francisca is the Charles Dana Chair in Human Cancer Genetics, Professor of Computational Biology at the Dana-Farber Cancer Institute in Harvard. She does some amazing computational work she's going to talk about today.
Thank you, Forrest. Oh, the expectations are high. It's a great pleasure to do this with you, Forrest, and I'm so excited that everybody's here for our symposium. The way I set this up today is to show you a few things that we have done with the goal of inspiring and inviting collaborations across the different data sets and approaches that we have within the center. Here are the disclosures. We've been focused on trying to understand evolutionary dynamics of human cancers using computational approaches. And so we've been interested across the years in both initiation and progression, both spatial as well as genomic and epigenetic, as well as the selection pressure that treatments administer to tumors to create this bottleneck that, unfortunately, in many cases, can be overcome when relapses occur. And so we've been interested in quantitatively characterizing these different stages of disease progression across different cancer types, both within patients and across patients. And so what the lab does is to have a very data-driven approach where we start with either in vitro or in vivo patient data sets to understand what the structures and distributions within these data sets are, to then come up with integrative analysis pipelines with the ultimate goal of creating predictive computational modeling frameworks with which we can ultimately impact patient care. And so the example that I want to show today is something we started years ago on GBM, but that has led to many follow-up investigations that are still ongoing and that are also part of our CSPC going forward. And so in this quest of investigating the causes and consequences of tumor evolution, we were interested in this particular example to come up with a quantitative description of how glioblastomas respond to treatment, different combinations of treatment, but particularly radiation therapy, with the ultimate goal of designing novel clinical trials that would be predicted to outperform existing fractionation and combination schedules, particularly focused on existing agents, but of course also branching out into other approaches. And so we're particularly excited about the BTC work and what Nino has just shown us. And so GBMs don't need an introduction at this point anymore, but what we set out to investigate was the question whether different radiation fractionation schedules might help maximize efficacy of a given amount of radiation. And so standard fractionation is 2 gray a day, 5 days a week for 6 weeks. But many investigators have investigated alternative fractionation schedules, including hyper and hypofractionation. Over the decades, the different fractionation schedules have been investigated. No significant differences were found in survival depending on the fractionation schedule. All that seemed to matter was the total amount of dose, which as the first line treatment is 60 gray over 5 weeks. We thought that maybe there was an opportunity to further improve and further investigate alternative fractionation schedules, given that we now have a better understanding of intra and intertumor patient heterogeneity, thanks to the TCGA and single-cell RNA-seq studies and other studies, such as the one that Antonio just showed, which then enabled novel, more predictive mouse models to be built, which then would enable computational models based on quantitative descriptions and measurements obtained from these mouse models. And so one of the early introductions of TCGA, of course, showed different subtypes of the disease that are partially overlapping with what we just heard from Antonio, but suggested that different subgroups of glioblastomas that are driven by specific molecular abnormalities. And that enabled Eric Holland's lab, who we started collaborating with when I was still at Sloan Kettering, to build a mouse model which is based on the ARCAS-TVA system, which is an avian virus, the receptor to which is engineered into specific subsets of cells within the mouse brain, including nestin-positive cells, but also differentiated cells marked by OLIC2, which is a cell surface marker staining differentiated glioblastoma cells. And so those cells that have been engineered to express the receptor can then be transfected with whatever molecular abnormality that this avian virus was engineered to deliver. And so you can build specific mouse models through this system that have stereotactic injection of the correct or mutation of interest or overexpression of a soluble factor of interest at this specific time, also sequentially, and within a cell type that you're interested in. So we started looking at the PDGF-driven subtype of glioma, where we deliver PDGF-A overexpression through RCAS-TVA to nestin positive cells in the mouse brain. And those histopathologically plus genomically look very similar to human GBM. And we can then use this mouse model to very carefully characterize the microenvironment and the dynamics of radiation response of different subsets of cells within this mouse model. And so if you just stain for H&E, for instance, you can see that you have endothelial cells here in the middle of this glioblastoma. Cells line the endothelial cells all around. If you stain for nestin, which is the stem cell marker, which I'm so grateful that Nino already introduced this, it looks as if in this mouse model, these stem-like cells that stain positive for nestin line the endothelial cells, so more closely localized at the center of the tumor lining the blood vessels, while the OLIG2 positive cells, the differentiated tumor cells, are more distal from the endothelial cells. And this seems to be supported by nitric oxide signaling that originates from endothelial cells and maintains these stem-like cells, at least in this mouse model, in an undifferentiated, more radioresistant state. And so this is important because we can then quantify, using this mouse model, serially over time and over different radiation fractionations, how sensitive versus radio-resistant individual subtypes of cells are, and how the time course of radio response for different localizations and cell types looks like, which of course we can't do with human samples. And so this is why we set out to investigate this based on this particular mouse model in collaboration with Eric Holland's lab. And so this is a paper that was already published a decade ago, but where we used the quantitative estimates of this mouse model treated with different fractionation schedulers and different total amounts of radiation administered to these mouse populations at different time points, and then measured how many cells remain certain times after administration of radiotherapy. And that enabled us to build a mathematical modeling framework that is very data-driven, so directly derived from the radiation response that we observed within this mouse model, in which we have two types of cells, the nestin-positive stem-like cells and the OLIG2 positive differentiated cells. If we administer a dose of radiation to these cell populations, then a fraction of the differentiated cells die, a smaller fraction of the tumor stem cells die because they're largely radioresistant, as parameterized based on the quantitative measurements of this mouse model. And there's also a small proportion of these differentiated cells that can de-differentiate into this more radioresistant state. We then use this parameterized mathematical model to optimize over all possible dose administration schedules of 10 gray given during the span of 1 week. So just 1 week of the schedule it would be administered to humans. But in mice, we can't give 60 gray, so we optimized for 10 gray. And we came up with this kind of non-standard schedule where we give 1 gray of radiation Monday at 8 a.m., 2 p.m. at 5 p.m., Tuesday at 5 p.m., and so forth, which we then implemented as a large mouse trial, comparing this optimized radiation fractionation schedule to several other 10 gray schedules given all at once at standard fractionation, or a sub-optimized randomized control schedule that was predicted to not be significantly different from standard of care, as well as a 20 gray schedule that was administered at the standard of care fractionation. So 20 gray total for 2 weeks, but 2 gray every day, just double as long, so double the total amount of radiation. And what we found was that this optimized schedule, even though it only administers 10 gray total, almost doubles the efficacy of each gray of radiation administered, because it's indistinguishable in terms of survival benefit in this mouse model from a 20 gray standard fractionation, suggesting that there is some scope, at least based on this mouse model, that we could think about optimizing how we administer existing agents in the best possible way to maximize efficacy. And so The mechanism seems to be that we enrich for nestin positive cells through this fractionation schedule where we give an additional dose before the potential stem-like cells either have been eradicated or replenished from dedifferentiation of the differentiated RLIF2 positive cells. And so this characteristic, very non-standard fractionation schedule seems to enrich for these nestin positive cells, which are even more radioresistant, but prolong survival because they have a lower turnover rate. So it seems that the mechanism of maximizing survival given this non-standard radiation fractionation schedule is that we enrich for even more radioresistant cells, which in principle doesn't seem like the greatest idea ever, but given that it's not curable anyways, at least it prolongs survival, at least in this mouse model of the disease. And so there is supporting evidence that this might also be a mechanism in humans, even though this is not definitive. But there was an Italian trial for the Antonios where patients were biopsied before and after radiation treatment, and those patients that had more than median enrichment in nestin-positive cells had significantly longer survival than those that had less than median enrichment. So it seems as if this could be a mechanism that could also be at play in humans, but of course many more follow-up investigations need to be implemented. We then decided to design a clinical trial based on this in mouse validated schedule, where we are interested in comparing the efficacy, well, the feasibility, first of all, we're not at efficacy quite yet, of this non-standard fractionation schedule against the historical control trial, which is RTOG-1245, that administered 35 gray in 10 fractions to recurrent GBM. And of course, we started with recurrent GBM, even though this is more of a mouse model that mimics newly diagnosed GBM because it's easier to recruit and more feasible to implement in those patient populations. The practicality constraints were a maximum of 3 fractions per day and not more than 3 days total that have more than 1 fraction a day. And so keep in mind that those are very sick patients that have recurrent GBM and so therefore can't be bothered to come in every day for many, many fractions. And also therefore to reduce the number of fractions as much as possible. So we redesigned this optimum schedule to something that's not the global optimum, but almost as good as this optimum that we had identified in the mouse model before, but simplified with the hopes of being more clinically implementable and more feasible. And so this led to this clinical trial here, which is called MARS-GLIO, which stands for Mathematical Model Adapted Radiation Schedules in Glioblastoma. The study objective was to employ a novel mathematical model adapted radiation fractionation schedule in patients for glioblastoma. We enrolled 14 patients after translating this mouse model using a series of other clinical outcomes where clinical trials were designed based on preclinical evidence based on the same mouse model to see how predictive the outcomes of this mouse trial preclinical evidence would be for human disease. And we then reparameterized using some data from human patients to come up with this MARS-GLIO schedule, which looks like this. 2 weeks total. First week, 1 fraction every day at 3.96 gray, 24 hours apart. And then the second Saturday, Sunday off. The second week, the first 2 days the same as the first week. And then the last 3 days, 3 fractions each at 1 gray each with a 3.25-hour interval in between. And that by this validated mathematical model would have predicted to, if scaled for efficacy, would lead to superior outcomes. But here we are only powering for feasibility, which ended up being 14 patients with the assumption that within 1 hour of these 3 fraction a day time slots, patients would be adherent to the schedule, and within 3 hours of the single fraction a day, patients would be adherent to the schedule. That's how we do the power calculation for 14 patients. So here are patient characteristics for the patients that we enrolled, median age 53, 4 women, 10 men, and the rest we can look at as well. Progression-free and overall survival look like this. It's not a randomized clinical trial, right? But it's compared to a historical control arm of the RTOG-1245, so we don't have a randomized progression-free and overall survival. But in a Cox regression, the progression-free survival is significantly longer. So the hazard ratio of death is smaller as opposed to this historical control trial, but of course, take it with a grain of salt because it wasn't randomized. We don't see the significant difference in overall survival. Here's the patterns of response in progressive disease across our 14 patients. We had some that maintained a partial response for a pretty long time. Of course, also patients that progressed very quickly. The most interesting finding were the patterns of failure within these patients, where in the external control arm, the RTOG 1205 trial, we had a distant failure rate of 16%. So almost everybody who recurred within the field of radiation. That was the planning target volume of radiation from the beginning, versus in our trial, we had out-of-field recurrence in the majority of patients. And this is a very significant difference where we see in just 2 cases recurrence within the planning field. Most of them actually recurred outside of this field of radiation, which suggests to me that we are far from having efficacy readout because it was only 14 patients. But we do see that we have significantly steered the evolutionary dynamics of these tumor cell populations in a different direction, because it looks as if within the time span of survival of these patients, we have sterilized the radiation target planning fields, and recurrence actually arises outside of that. Now, the question, of course, is what to do about that, given that we can't give that much radiation to the entire brain. But I think the next step is then to look at efficacy, which we are now talking to Brigham and Women and Dana-Farber radiation oncologists that also ran our first clinical trial to actually expand this to 40 gray. There's another RTOG trial that administered 40 gray to the recurrent setting, because the hope would be that, of course, the higher we go, the more efficacy we will have, and the lower the enrollment numbers are going to be in order to see statistically significant outcomes. And so the standard schedule here that we are going to compare to is 2.67 gray in 15 fractions. So this is now given over 3 weeks, 5 days a week. And then the same fractionation every day for the historical control, which gives us this constraint of BED equivalency of 93 gray that we can administer in this optimum schedule. And this is all work in progress by Chris, who is here, where we're now looking at optimized schedules for this 40.05 gray schedule, where we have already identified several candidate schedules that are predicted to significantly outperform this standard fractionation schedule. Some of these schedules look like, those that are optimized look like the de-escalating schedules are best, where we start with high dose and then later on have lower dose, but more frequent fractionation schedules. And if you look at the normalized volume change of those schedules, as opposed to the standard, again, this is going to be the historical control, then we have a significant reduction in normalized volume change over those optimized schedules, really non-significantly different over the different optima that we have found, both in terms of total cell number and in this nestin positive subset of these more radioresistant cells. And so the sample size calculations based on the hazard ratios that we scale from the mouse model that we also used to come up with the sample size calculations for our first clinical trial suggested we need maybe around 75, maybe to be safe, 85 patients to enroll in this patient trial. And so we're working on implementing this currently. If anybody's interested in the multi-institutional trial, please let me know. That would make accrual much faster, but of course the bureaucracy is a little bit more complicated. This is in combination, in collaboration with Shyam Tangatory and Daphne Haskogan at the Brigham and MGB. The next step then is concurrent therapy, where we started thinking about temozolomide and radiation just to address the newly diagnosed setting in the beginning. For that, we need to move to a spatially explicit model, because unlike radiation, which is a field effect, so it doesn't really matter where cells are localized, how much radiation exposure they get, as long as they're within the target planning volume of radiation therapy. Now, temozolomide diffuses away from the blood vessels, so therefore the exact localization of the tumor cell population matters for exposure of the concentration of temozolomide that's present within the system. So what we have here is a cross-section of an agent-based spatially explicit model that has these tumor blood vessels coming out at us like that. So the endothelial cells are the red cells, and then our stem-like cells, the nestin-positive cells, are blue, and the differentiated cells are green. And so according to the rules that we again parameterize based on this mouse model, we can now run forward stochastic simulations to identify optimum dosing schedules in this increased setting. That might be a little smart to read, but where we have a radiation response and in addition, a chemotherapy response that can either kill tumor cells or if not, they age by 1, so that we can keep track of not only the spatial organization, but also the age structure within the cell population. And so what we did there was both optimize radiation schedules, keeping the temozolomide administration schedule constant, but also keeping the radiation schedule constant and then optimizing the offset between temozolomide and radiation administration at standard fractionation, thinking that that might be actually easier to implement as a clinical trial. And what we found here is that the optimum offset between temozolomide and radiation given at standard fractionation would be, for mouse PK parameters of temozolomide, 41 minutes, for human parameters, 57 minutes before radiation temozolomide should be given. And that also validated in a mouse trial where the suboptimum was given, temozolomide 8 hours post-radiotherapy, as opposed to the optimized where temozolomide would be given 41 minutes before radiation. And that leads to a significant survival difference. So that now is the question that we've been discussing with Chen and others, whether we should implement this as a clinical trial as well, and what other evidence there is just by natural variation within different radiation oncology groups, whether they recommend temozolomide to be given before radiation or maybe at bedtime or at a non-recommended time, and whether we could potentially do retrospective analysis of these data sets to see whether there would be difference in outcomes depending on when temozolomide is given. We've also been working on optimum combinations with radiotherapy and PARP inhibitors in combination with one of my previous postdocs and also Anthony Chalmers' group in Scotland, and also collaborating with Chen about the ATM inhibitors that he will talk about later today, and then we will also discuss tomorrow during the site visit a little bit more. And then the last aim is to look at patient specificity, where the goal is to adapt these models using patient-specific either biomarkers or ex vivo tumor assays that could be used to then come up with a patient-specific optimization for each individual patient that can take into account also what these patients have already received up to the point of optimizing radio combination strategies. For instance, whether they're already at 60 gray or maybe they're already at 75 gray, and so we only have maybe 35 or 40 gray left to optimize. We can also take real-world constraints into account, for instance, transportation issues, or what if the bus is late, should we update the schedule later in the week? And so hot off the press is something that's also Chris Gasser's work that I wanted to show here as an example of how we can use in vitro data sets to try to come up with personalized optimizations. This is not specifically for GBM, but derived based on cell lines of a variety of different cancer types. But the question here is, what if irradiation actually differentially impacted patients with respect to how migratory cells are that have been exposed to radiation? And so if that's true, that there might be a subset of patients that has a positive correlation between dose exposure and migration kinetics, then that's an interesting trade-off between how much radiation we should give to maximally inhibit the tumor cell population, but also maybe induce migration, which could be related to metastatic behavior. So that might be a dangerous thing to do. If you have a patient where there's a negative correlation, then maybe that's a less important consideration, because then the optimum for depleting the tumor cell population would also be the optimum for actually minimizing migration. And so we could imagine that if we had measurements such as those for a diverse patient population, that we could come up with a patient-specific optimization schedule to create individualized treatment plans for each individual patient. And so in collaboration with Galit Lahav's lab at the HMS Systems Biology Department, we characterized a number of different cell lines in terms of their migratory behavior in response to radiation exposure. Here on the x-axis are the different gray of radiation that were administered to these in vitro cell populations. And then we measure speed of displacement. And this is Brownian motion. So it's not a directed migratory behavior. It seems to be just random migration. But you can see that in some of those, this is a lung cancer cell line, for instance, there's a positive correlation between radiation exposure and migration. which is interesting. So then we built this spatially explicit model based on the model that I showed you before, where we can now also take into account whether the cell population migrates outside of the target planning volume as defined for the radiation dose administration schedule. And we can then count how many of these escaped tumor cells we have. The goal being that we now have another goal for the optimization to minimize the escaped tumor cell populations if there is a direct correlation between this escaped tumor cell population and potentially migrate, like distant metastases, or just in the brain, as we've seen in previous talks, movement to other areas that would then lead to out-of-field recurrence. And so for one of these cell lines, for this lung cancer example here, we can identify an optimized schedule, which is a de-escalating schedule over here, which, as opposed to the standard schedule, very much decreases both the escaped tumor cell count and the primary tumor cell count. So the prediction would be that would lead to better survival and also lower rates of distant metastasis if this migratory behavior is really predictive of distant metastatic behavior. And we can then read out for these different schedules that we have created here, the overall survival for the different patient populations. And then finally, using this in silico clinical trial data set of 1,000 patients, we can then, that are parameterized using distributions of radiosensitivity and also these dose speed coefficients as identified based on this in vitro cell population parameters, we can then use this in silico longitudinal data set, which only gives us, based on these input parameters, the number of the primary tumor cells and the escaped tumor cells. And we can then train a transformer on this longitudinal 1,000-patient data set to back out the parameters that we can't measure in patients, right? But that we know what the gold standard is in this patient cohort because it's simulated. We can identify the migratory and radiation response parameters based on this machine learning approach, and this is work by Manuel Schürch, who's also here, with the ultimate goal then of having a proof of concept of imagining a patient population where we have longitudinal measurements, for instance, of the primary tumor, and some metric that would be correlated to these escaped tumor cells, for instance, proportion of CTCs in response to radiation. So if we treat a patient with standard fractionation, for instance, for the first, week or two of radiation, and measure longitudinally what these volumes are, we could then use this pre-trained transformer to identify the parameters of that individual patient, of the parameters that we can't measure in vivo, and then optimize, given those parameters, a patient-specific optimum schedule for the remainder of the treatment administration schedule. And so that's just a proof of concept of how we are thinking about personalizing treatment. The problem, of course, is that it's very hard to get access to data sets like that, but that's why I'm so happy that you are all here, and maybe you have some data sets that can help us solve this problem going forward. And with that, we would love to collaborate if you have any thoughts or any ideas for this. Big shout out to the lab. Big shout out to the CSBC, that's been super fun, and also all the collaborators over the years that have enabled us to do this. Thank you.
Questions? Nina.
I've always been very bad at math, so I'm happy somebody understands it. But two quick questions. One is a comment. We think that having more, enriching for stem cells is actually bad. We want to enrich for differentiated cells. But you show it's the opposite, actually, really. And what, are you surprised by that? And if you want to comment on that. And the second question is, I was thinking that the other FDA-approved treatment where this may be really helpful is, I know we don't use it that much at Dana-Farber, is the TTF fields. Because there it was sort of like empirical that patients have to use it for 18 hours at a certain alternating current. It just seems to me that that's just like radiation, that's a device that's giving you a physical something. I'm just wondering whether you've been thinking about adding that in your model.
Yeah, that would be great. We'd need some data to parameterize models like that, so I'd love to discuss where we could get access to some of those. The first question, yep.
Not at Dana-Farber, we don't do it very much, but there are other places that do a lot of it.
Like which places?
I'm trying to think.
Okay. Yeah, that'd be
a
great.
lot. A lot of places around the. At Mayo Clinic, don't you use a lot of TTF?
We use
use it, but it's an offer to everybody, but it doesn't actually get used that often. I mean, obviously, you think Northwestern.
Probably Northwestern would be the best place, right? There are a lot of places they use it.
Yeah, that would be great. It's best if we have detailed quantitative longitudinal data that we can use to parameterize these models. So sometimes the human cases are not detailed enough because maybe just given at one fractionation schedule or one combination therapy schedule. But that would be interesting. And your first question, totally agree, and it surprised me. I think the reason is that they proliferate less quickly. So if you're only interested in delaying the time of the progression of disease, right? Then that might be the way to get there. But it depends on the exact interplay between the growth rate of the differentiated and the nestin positive cells. In this mouse model, it looks as if the nestin positive cells grow less quickly, even after radiation has been discontinued. And that's the mechanism here, it seems. But of course, that's totally, right? Probably mouse model, maybe even PDX line specific. And I wonder whether we could measure that somewhat to then, again, come up with a personalized schedule depending on what the ratio of growth rates is, both before and after radiation.
It's great work. I'm just sort of wondering if, similarly to you showed a bunch of data saying that we had these optimized schedules for tumor response. Have you looked at treatment-related necrosis, radiation necrosis, and other things with this? And you know, there are these two things that you're balancing back and forth as you change the schedules.
In the human schedule or in
well, either, but in
yeah.
Patients particularly, of course.
Yeah, so I mean, for the humans, the only readout we have is, I guess we have some patient characteristics at the beginning, and then the readout is progression-free and overall survival. But the trial that we're designing now will also have longitudinal MRI, maybe even with some of the more modern methods that we have at the Farber now that could give us a little bit of functional readout as well. And I think that would be much more interesting because then we could even incorporate maybe some diversity measures from radiomics readouts. But I think at the end of the day, it's a problem of getting access to enough quantitative longitudinal information. It's even hard for us to get the MRI for everybody in GBM that's been seen with a longitudinal readout, just to see whether the ratio of rates that we estimate from the mouse model are correctly scaled for the human case. So for us, it's been a little bit of a problem of getting access to clinical data. If anybody has some, I would be very happy to take a look.
Hey, Francesca, really nice talk. I guess the question I had was about the generalizability of the model built on the GEM. Generally, when we think about 10 gray given as a single fraction versus 10 gray given in 5 fractions, a single fraction is going to be much more effective just because of the linear quadratic nature of the survival curve. But I think if I understood from the data, there wasn't a huge difference between a single fraction and fractionated. And so I guess I'm wondering, like, how do you think about that as far as generalizing to a population of patients?
Yeah. This one here, right? Yeah, I think that's a good question, right? And that's partly why we're interested in looking at all of your data to see whether we can, right? The goal for the first ATM, the AZ inhibitor project, right, is to take this model parameterized on that mouse model, update with your data, and see whether that's predictive for a newly designed mouse survival study. If that works, I think then that's the proof is in the pudding that is generalizable. We haven't tried for many other mouse models apart from this one here. We have a few other RCS-TVA-driven systems with different molecular alterations. For instance, we tried this with non-INCFIR null backgrounds and just PDGFA overexpression. And that seems to have similar responses. But I think at the end of the day, the question is also going to be how much do we actually believe in these subtypes, which is one of the questions I wanted to ask you, Antonio, right? We have all of the TCGA-determined subtypes, and then we have your subtypes, and then we have Brad Bernstein's data that shows that everything is everything anyways. So I wonder a little bit how, right, if we have to have a bespoke solution to each individual patient's tumor, or whether it's just a very noisy system and the subtypes are maybe less important than we thought in the beginning. And I think that will also be part of the answer to this question here. And I don't think we know that yet. Or at least I don't know that yet.
Thank you, Francisca. That was great. I was curious about the observation that you saw when you saw recurrence in the clinical trial. It was typically outside the field of treatment. I was wondering could you go back to the pre-treatment MRIs and examine those regions to see if there's any evidence of lesion or abnormality that could have been spotted prior to treatment and maybe then actually treated?
Yeah, they didn't look very carefully at all of them, but they checked a few, and there didn't seem to be any evidence. But I guess that doesn't surprise me all that much either, right? Because if you look at, right, one of the Antonio's talks, and also this mouse model here, right, where if you actually label the tumor cells, they're already over the entire hemisphere anyways, even though, right, the contrast enhancing region would be a small subset of that. So I think the story